Hello, I'm Will. Welcome to ResearchPod. Now might be a very good time to get into virology. In a post-pandemic world, governments around the globe are looking to get ahead of whatever the next threat to public health might be. On the other hand, it might be a bad time to already be a virologist. There are hundreds of diseases impacting millions of people that aren't getting the same treatment, and the traditional pace of progress could seem very slow to outsiders. Dr. Matthew Reeves of University College London joins us today to talk about CMV, one of the herpes family of viruses that has likely infected billions of people just since the 1970s, when attempts to make an effective CMV vaccine were first initiated. His research could provide a valuable step forwards for stopping the spread of infections between people, even between cells, and may open the door for wider-reaching breakthroughs across all virus research. Joining me, Dr. Matthew Reeves. Hello.
Hi, Will. Hello.
Thanks very much for joining us. Could you tell us a bit about yourself, your research, and what brings us here today?
Sure. So my scientific career really gets started with my undergraduate degree in molecular biology. So that's understanding how cells in the body work. And that was really my interest. And then I went on to do a PhD, and I'd never done any virology at all in my degree. And I was thinking, how do we understand how does the cell work, and why not ask an expert? And the expert is a virus. Viruses have worked out how our body works, how our immune system works, how all our cells work, and they're users for that benefit. And so I went on to study virology at Cambridge for my PhD, and essentially I was a cell biologist working with a virus to understand how the cell works. And the virus that I was interested in, I'd never heard of, which is cytomegalovirus. And I think that just shows really that it wasn't the virus that drew me to my PhD. This is a tool to understand something that I was interested in, which was cell biology. As I learned to understand this virus, and then as we'll come on to discuss, I think later, this is a really important virus for medical reasons. But what I was trying to understand was, in my PhD, was how does this virus infect people and live with us for life? And really that was my PhD. And then I moved on to, as a standard scientific progression, as a postdoctoral position, and I stayed at Cambridge. And this was a slightly different question I was asking. I was asking, how does the virus keep our cells alive? Because the virus wants to keep our cells alive so it can replicate and make new copies of itself. And then I decided I fancied something a bit different. So having been in Cambridge in the UK for 7 years, I won a fellowship to go to Novartis in the US. And I moved to Cambridge in the US, Massachusetts, so same name but different country. And I spent 3 years there, and the idea was to go to Novartis and be given really free rein to work on CMV, but it's something I found interesting. And I went to work with Teresa Compton, who's a really big expert in cytomegalovirus entry. How do viruses get into cells? And so I took my expertise in understanding what the virus does when it's in a cell and allied it with Teresa's work on understanding how does the virus get into the cell in the first place. And I think science is full of serendipity to some extent. So at the time, Teresa was a big expert in a protein, which is important for virus entry, which essentially became a focus of study later on for the vaccine. And at the time, that wasn't why I went there. I went there just to try and understand how does this virus get into a cell. As always in science, you move around a lot. So we decided to come back to the UK, and that's partly because we were starting a family, and we wanted our family back in the UK, so grandparents wouldn't hold it against us when we were living so far away for all our lives. And so we came back to Cambridge, and I got a Medical Research Council fellowship, and this was really my first opportunity to start my own laboratory, which has underpinned really the work that I'm working on today. I guess it's three things. One is that long-standing interest in how this virus can infect us and infect us for life, which we call latency. Then there's also the aspect of how does the virus get into cells and what does this mean for immune responses, and particularly antibodies and how they protect us. And finally, what does this mean for developing a vaccine, which has really become a goal of mine in the last few years since I met up with Paul Griffiths at University College London, where my laboratory is now.
Well, everyone listening to this is going to have something of a crash course in virology over the last couple of years, so there's going to be some parts of what we're talking about today that may chime with recent history. But to set the scene about CMV in particular and what sets it apart from other viruses and other health complications, could you tell me from the top down what is, like, even the size of the virus particle, what is its structure, what does it do, and what does that mean for health? A little bit of the etiology and what this means for healthy bodies, unhealthy bodies, and kind of how disease comes around.
Okay, so CMV is one of 9 herpes viruses. And so people will have heard of herpes viruses. And the thing that people know about herpes viruses is they're for life. So once you're infected with a herpes virus, you have it for life. And the herpes viruses that people know about are the virus that causes cold sores, herpes simplex virus, the virus that causes chickenpox, which is the varicella zoster virus, and then later on can cause shingles. And people will have heard of glandular fever, which is also a herpes virus, which is Epstein-Barr virus. People haven't heard of cytomegalovirus, but it's part of that family. And like all the other herpes viruses, can infect us for life. And what they are, they're actually very large viruses. And so if we take, for example, HIV, which is a virus that everyone's heard of, its genome is about 10 kilobases in size. Cytomegalovirus's genome is around about 240 kilobases, so it's bigger. It's one of the biggest viruses that actually infects us. And what that means is this virus has, comparative to other viruses, has lots of proteins that it expresses, which are important for infection. What it essentially is, it's a piece of DNA wrapped in some proteins, which we call a icosahedral capsid. Around that are some proteins floating around, which are the tegment proteins, and these are proteins which will enter with the virus and start to take over the cell, even before the virus has started expressing any of its actual genes. It delivers lots of what we call preformed proteins. And surrounding all that is an envelope, and it's a lipid envelope. And in that lipid envelope, there are lots of proteins embedded in that. And they're the proteins which allow the virus to find and enter its target cells. And perhaps one of the most famous proteins not in CMV, obviously in SARS-CoV-2, is the spike protein. Everyone's heard of the spike. And that's the protein which is embedded on the surface of the virus and allows it to infect cells. Now, CMV does not have spike, but it has proteins which have similar functions, of course. And so that's the virus. And does it cause disease? Well, if you're healthy, it doesn't generally. Your immune system is really good at controlling this virus. So we don't see disease with this virus in healthy people. But what we cannot do is eradicate the virus from our body with our immune system. So this virus can establish lifelong infections. And that becomes a problem because there are situations where we become immune compromised or immune suppressed. And so our immune system stops working. And so in the 1980s, the HIV AIDS epidemic, the phenotype of long-term HIV infection is AIDS, which is acquired immunodeficiency syndrome. And what would happen is their immune system was being essentially wiped out by HIV. And this left people susceptible to herpes viruses, for example, as well as other infections. And so they will often see herpes virus infections in these individuals, which will end up killing them. The other major population we're interested in now is our transplant patients. And so when people have a bone marrow transplant or a solid organ transplant, so a kidney, liver, lung, etc. Of course, what we are doing is we're taking something which our body would see as foreign and putting it into that person. And so our immune system naturally will attack it, that incoming organ. So what we have to do is immunosuppress people. And of course, by immunosuppressing them, we're also removing that immune control of the virus that's living in those people. And so the virus now can replicate uncontrollably and cause disease. And it can cause disease all over the body. So it can infect the liver, kidneys, eyes, brain. And so we see disease in all those organs, depending on where the virus is. The final population that we're really concerned about, and perhaps where CMV or the drive for a CMV vaccine comes from, is in congenital infection. And so the virus will infect a mother-to-be who is pregnant, and the virus itself doesn't cause disease in the mother-to-be, but if the virus transmits to the fetus across the placenta and infects the fetus, it will cause severe disease, so mental retardation, child might be born with deafness, with blindness, and essentially the child is ill for the rest of its life once it's born. And that's where the problem lies. It's around about 1 in every 150 babies born will be born with a CMV infection. So this is more than rubella, and rubella was a major problem in congenital infection. About 20% of those babies will have evidence of disease, and that's why we need a vaccine against this virus.
It's inescapable, I suppose, but I can't help but come back to vaccine development that we've seen with the COVID vaccine in the last 4 years or so. Should really be keeping count because I've been saying last 2 years, last 3 years, and time's getting away from me. In recent history, we have seen that rapid vaccine development and rollout. And I suppose there's going to be people out there who say, well, if there's 1 in 150 births for children who are going to be affected by this, why haven't we seen that same rapid rollout, that same massive development of vaccine effort and funding? Why isn't it like that for every disease?
And it's money. Well, it's money and political will. And the drive for the COVID vaccine was very unique. We had a situation where we had a population which was naive. We did not know the risk of the infection. We had to control the infection. And if we look at what happened in that situation, there was a gamble made. And the gamble was that the vaccines against SARS-CoV-2 will work. And so they started everything at the same time. So right through from vaccine design, vaccine development, through to production and scaling up. And because all that was happening at the same time, it was successful. Now, of course, if that vaccine had failed, then we're not sure what plan B was. But fortunately, it didn't fail. But actually what happens in reality is we can't do that for every virus. We can't just throw 150 million, 200 million pounds at it straight off and ask the question, can we do everything all at once? And normal vaccine development takes time. We have to go through these stages, what we call milestones. We have to say, well, it works in this system, therefore we think this is important, so we'll go to the next system, and then it can take 5 to 10 years. That's the first reason. The second reason is what are you trying to vaccinate against? And not wanting to shout out how CMV is fantastic from a point of view of immune evasion. This is a virus that has learned to live and evade our immune system. So if we go back to think about what a vaccine actually does, what a vaccine does is it tells or educates our immune system, should I say, what a pathogen looks like before it sees the pathogen. And so the immune response is faster. And invariably that works, that's protective. And that's what we try to do. The problem we've got is that CMV can infect and reinfect people who have an immune response against CMV. So whilst it doesn't cause disease, it can reinfect people. And so the hypothesis is actually we need to do better than nature with CMV. And I think that's why CMV vaccine progress has not perhaps progressed at the pace that SARS-CoV-2 has. And lots of vaccines have been tried, and we get immune responses, but they just don't reach a level of protection required to stop infection and disease.
Well, you mentioned the milestones of development there. What has the timeline for CMV vaccine development looked like so far? Have any milestones been met in any particular kind of systems, as you say?
CMV vaccine development is quite interesting. It's followed, I think, quite a standard course. So it's back in the 1970s when they really first started to think about CMV vaccines. And they took the approaches that have been successful with things like polio. Can you make attenuated viruses? So a virus that will infect people will not replicate and cause disease, which is often what we used to do for some of our earlier vaccines. They tried that. They got immune responses, but the immune response was not sufficient to be protective. So then people said, okay, so let's take bits of the virus, bits we think are important. So bits that we have seen very large immune responses against. And if we can make those responses, are they protective? And they've done it against major proteins, including glycoprotein B, which is what we're interested in as a vaccine. And they've all had some elements of success. So they've had some levels of protection, but what we're looking for is around about 70%, 75%. That's our benchmark for protection for a vaccine to be licensed. And to date, the best performing vaccine has achieved a success rate of around between 40 to 50% protection. So what we're trying to understand is why did it only achieve that partial protection, but more importantly, what aspects were good about that vaccine, and can we take those bits and make a new vaccine have those bits but actually get rid of some of the other bits which perhaps weren't so good. And so we've tried different strategies, and perhaps the most recent vaccine trial that's happening now is with Moderna, who are using their RNA technology. So the same RNA technology that was used to generate the SARS-CoV-2 vaccine, they're using RNA technology now against CMV proteins, or CMV targets, should I say, which again, the idea is can you generate immune responses, particularly antibodies, which are protective against this? And we'll await those data. We're still waiting to see whether they worked. I think RNA technology has perhaps changed vaccinology quite a lot, because now we can make vaccines in weeks rather than years and test them.
Thinking of those years, you say vaccine development started in the 1970s, with even just for the neonatal infection of 1 in 150 children born having CMV infection. To think about the time and the breadth and the scale of the infections through the 80s and 90s with the AIDS crises, the health toll of CMV infections, that is such a health burden for those past 40, 50 years.
It is. I was thinking about this because we always think in percentages. So I looked up the number of new births every year since 1970s. It's around about 3.5 million, 4 million new births every year. If you scale that up 50 years, that's 200. I won't get my numbers right now. 200 million.
Sure.
Doing the maths in my head very quickly. It's a lot of births, right? And this is a virus that's probably infecting. There's a seroprevalence around about half the population infected this virus. That's a lot of new infections, as well as people who perhaps were not carrying the virus before 1970 who've acquired the virus since. This is infecting a lot of new people. But if you just think of that from birth and those numbers I gave you before, if 1% are infected with CMV and have disease, that's a problem in itself. Then over time, these people will acquire the infection as they live. So I would say about half those new births are infected with this virus. And you're right to highlight that. And back in 2000, this is the US Institute of Medicine sat round and decided what did they think were the highest priority for a vaccine. So this is a conversation that has been had. And there was 3 viruses at the time which I think were selected for highest priority. HIV, which is always on the list, hepatitis C, at the time, since it was 2000. And actually, we've got some really good drugs against hepatitis C now, but I think a vaccine would still be desirable. But the other one was CMV. So CMV has been recognized as important for vaccine development.
Which leads us on to your work and your research, and the most recent publication from the Nature Communications paper on how the virus moves through the body, propagates itself in that infection. So if you could set some of the stage here, a little bit of the background information.
Absolutely. So it's actually very useful to go back to what the vaccine was and how it was tested. And so the principal component of the vaccine is a protein called glycoprotein B. So it's a modified version of this protein called glycoprotein B. So glycoprotein B is I don't want to say it's equivalent of spike for SARS-CoV-2, but it has a similar role. It's important functionally in that it acts as the fusogen. So what it means is that the virus can bind to the surface of the cell and enter the cell. It's one of the ways the virus gets into the cell, and it's essential for virus entry. And this is why this protein has always been included, I think, in vaccine preparations. Because the hypothesis is, of course, if you stop the virus from getting into a cell, it can't replicate. And that makes sense. And so in order to stop a virus from infecting a cell, what you generally need is antibodies. And this is how, of course, how most of the vaccines we know have worked. They've produced antibodies. And what we call those antibodies are what we call neutralizing antibodies, because they neutralize the virus before the virus can infect the cell. And so Paul Griffiths, who was a colleague here at UCL and the Royal Free Hospital, was working with our transplant teams. And so what they did was they vaccinated people waiting for a kidney or a liver transplant prior to transplant, and then allow the transplant to proceed and ask the question, how protective was the vaccine? And they showed the vaccine was protective for around about 50%. The assumption was it was all about neutralizing antibodies. And that was supported because what they observed is that people who had more antibodies against GB following vaccination did better post-transplant, which makes sense. They've got more protective antibodies. However, when we went and started to look for neutralizing antibodies, so these very specific antibodies which will bind to the virus and neutralize infection, we couldn't see any. And so we were a bit stumped because what we think of always as the correlate of protection and is the mechanism of protection wasn't there in these patients, but we knew they were protected. We could see they were protected. So we thought about it, and antibodies can work in more than one way. And what we also know is that viruses, not wanting to be anthropomorphic, viruses don't like neutralizing antibodies. They're a problem for them. And so what a lot of viruses which infect people like herpes viruses, and actually HIV can do this as well, is that once they're in an individual, once the virus has infected somebody, what they can do is actually hide from those neutralizing antibodies. And the way they do that is they never actually ever leave the cell. So they'll go into a cell initially and replicate. But rather than release the viruses outside of the cell, which would then expose them to neutralizing antibodies, a strategy that they've developed is a way where they see a neighboring cell, which is not infected, they fuse the two cells together almost and create a little tunnel through to the new cell. And what that means is those neutralizing antibodies can never ever see the virus. So even if you had neutralizing antibodies, they might not be protective. And what we know for CMV is that this is how the virus grows and lives in us for the most of the time. It goes from cell to cell. And so this started to make sense because we know that actually the virus is very rarely cell-free in an individual, and we couldn't detect neutralizing antibodies. So maybe it was another antibody response. Again, what we did is that we thought, well, what does the immune system do of someone who's been infected with CMV. So someone who's actually been infected with the virus, what does their immune system do? What does their antibody response against glycoprotein B look like? And they make neutralizing antibodies, but they also make antibodies against specific regions of glycoprotein B. So glycoprotein B is a big protein. It's got 906 amino acids. But within that, there are regions which are targets for antibody responses, and we call them antigenic domains. And in natural infections, so people infected with CMV, there are 5 being recorded. And we went looking for responses in our vaccine recipients, and we couldn't find them. So what this was telling us was that the vaccine was producing a different immune response in people. And is this why the vaccine was protective? We looked in more detail and asked the question, where do those antibodies bind on GB? And we found what the paper's about, which is AD6, which is a new antigenic domain. And so people who are infected with the virus rarely make antibodies against this region, but people who have given the vaccine do make antibodies. So the question is why? Why does the vaccine cause this response, but not the virus? To think about this, we have to think about why there would not be a response against AD6 in natural infection. And the reason for that is that AD6 is hidden. That region of the protein is hidden from the immune response. And the reason that the virus, we think, does that is because it does not want antibodies to target it. And the reason for that is because if it does have antibodies that target it, what do viruses have to do in the face of an immune response? We've seen this with SARS-CoV-2. They tend to have to mutate, and that's how they escape from antibodies. So we've seen that with Omicron, for example, has lots of mutations in spike, which allows it to escape from partially escape from antibodies. But the problem is, if the region you're targeting is absolutely dependent on not mutating, i.e., to function, it cannot mutate, the virus has a problem. And so its solution is to hide it.
For the phrase highly conserved, is that the...
That's the word. Yeah, it's highly conserved. And if you look in CMVs across, we looked at 300 different CMVs from different patients, it doesn't change. Amino acid sequence is exactly the same. And that's because it's not under immune pressure to mutate from antibodies because it's hiding it. But the difference between the vaccine and the virus, and this is serendipity, and a lot of science is about serendipity. The vaccine GB was slightly changed, and what that meant was it presented that region to the immune system, and so it could activate an immune response. What we then see is if we have antibodies which target that region, they can see GB in certain situations. They can't see it on the virus because the virus has hidden it, but there is another important place where this protein is functional, and that is on the surface of the cell. So when the virus infects a cell, it puts glycoprotein B on the surface of the cell, and that's part of that cell-to-cell connection that I talked about earlier. And GB is involved in that. And we think that the antibody sees GB in that context. And by binding to GB on the surface of the cell, it prevents the ability of that GB on the surface of the cell to make contact with the neighboring cell and to cause the tunnel I talked about earlier to perform, which the virus can go through. And we think that's how the antibody was working. I guess it's an evolutionary warfare. I think we've got a host is evolving to try and protect itself, and we've got the viruses. And I think sometimes what people have to bear in mind is the virus isn't necessarily evolving to cause disease. That is not an endpoint necessarily for a pathogen. Success for a pathogen is to exist and transmit, and that actually is probably the primary success criteria. If it can get in and survive and transmit to a new host, actually causing disease is not necessarily a good thing, unless that disease is important for transmission.
Thinking of AD6 and this tunneling and preventing the spread, then it's very tempting to jump right to the end and say, oh, so you've got a cure, you've got a vaccine, you've got the solution in your hands. Or is that being very preemptive?
I think it is preemptive. I think there would be a certain hubris as an investigator to think that there is only going to be one response important for protection from cytomegalovirus. And I say that because people have been working for 50 years trying to find a vaccine. I think if a vaccine was that simple and was one response, that's going to explain it. I think it's going to form part of response. And I think it comes down to is actually what do you want a vaccine to do? And again, we saw this most presciently with the SARS-CoV-2. Do you want a vaccine to stop transmission, or do you want a vaccine to stop disease? In an ideal world, you want a vaccine that prevents transmission. That's the ideal. In absentia of a vaccine that stops transmission, you want a vaccine that stops disease. And that's what the SARS-CoV-2 vaccines have done. They've stopped disease, really. But by stopping disease and reducing the level of viral replication, you can have an impact on transmission as well. So if you replicate less virus, you're less likely to transmit that virus. So that's why, whilst the SARS-CoV-2 vaccines haven't directly blocked transmission by bona fide measures, they probably reduce transmission by stopping the level of viral replication in people who are infected. And that's similar sort of idea with CMV. So you think about the vulnerable patient populations, it's the fetus which is vulnerable. So you've got two options. You could stop infection of the mother to be, or actually all you really need to do is stop the transmission of the virus from the mother to the fetus. And it may be harder to stop infection of the mother than to stop transmission of the fetus, because they may be very different routes. So the infection of the mother to be is cell-free virus, probably respiratory infection, whereas the transmission of the fetus could be a cell-associated virus. What we would say is perhaps what you want is a combination vaccine, and a vaccine which targets the cell-free virus and a vaccine that targets the cell-associated virus. I've also not mentioned the other really important arm of the immune response. That's the T cells. So antibodies are great and protectors, but actually long-term protection from cytomegalovirus is thought to involve T cells. And so these cells which target infected cells. And if we can harness that response in a vaccine, then that'll be even better.
Again, to draw a war analogy, it sounds like by stopping cell-to-cell transmission, you're blocking off reinforcements and cutting fuel lines to the front and all that.
Yeah, essentially slow the virus down. That's what you've got to do. You've got to slow its spread before the reinforcements arrive. And then I guess what we call a counteroffensive.
That sounds like, you say, would be the ideal to have something that does both of those. Is that research ongoing in development so far as you know? Where does this paper on AD6 and cell-to-cell transfer lead to next for your research and for the global effort against CMV and mononucleosis?
Yes, so we are developing it, and we're looking to work with partners to do the test. And the test is, does a vaccine which contains AD6 as a target work in our patient populations? And we're very fortunate here at the Royal Free Hospital because we have a fantastic transplant team. We do lots of renal transplants, we do lots of liver transplants, and they are very keen for testing new medicines. And so if we have a vaccine developed, we can take it straight through into phase 1 for safety, then phase 2 for actual testing. And can we protect our transplant patients? But again, I'll reiterate, what we see this as part of a larger vaccine strategy, which will probably encompass multiple key targets against CMV, which would harness both neutralizing antibodies, non-neutralizing antibodies, what we're talking about here, and also, I hope, T-cell responses as well. I think we're going to need everything against CMV because CMV has an impressive arsenal of immune evasion genes. And so of that, remember, right at the start, I mentioned it's this very large virus, 200 kilobases of DNA, has 200 proteins, which is a lot for a virus. HIV, 7, 8, 9 proteins, the HIV, so they're much smaller. They do things with a lot less proteins. And so CMV has a lot of proteins, and I would say now about 50% of those proteins are dedicated to immune evasion. It's really going to war, to use your analogy, against the immune system in that respect. And I think it has to, because if you look at somebody who's infected with CMV and you look at their immune response, if you look at possibly how many T cells, so all those infections they've ever seen in their life, and you look at how many of their T cells recognize CMV, it can be upwards of 20%. So our immune system dedicates a lot of its resources to combating and controlling the cytomegalovirus. And that's because the virus itself has all those immune evasion genes. There's a battle going off all the time.
To look in the short term more than the long term, with ideally there being that vaccine development, are there any upcoming publications that might be coming from your team that anyone reading this paper could say, okay, now I want to know more, I'm going to follow up with what's going on at the Reeves lab, or any ongoing projects, conference appearances? Is there anything that you'd like to plug for, the next couple of months here?
I think the exciting thing that's coming from the lab for me, and hopefully will be coming out, is being presented at conferences, but actually is hopefully will be coming out soon as a publication. We're just putting it together. As I alluded to before, there are 9 herpes viruses that infect humans. And what's interesting, they all have glycoprotein B. And actually, they all have this region, AD6, in their glycoprotein B. Sequence-wise, it's not conserved, but from a structure point of view, it is very conserved. And antibodies can recognize structure as well as sequence. And we have some, I think, quite exciting data that says that these antibodies work against other herpes viruses. So if you're going to cure one, cure them all.
Might as well while you're there.
That's a long way off, but that's quite an exciting story that's coming out from the lab at the moment.
Very much look forward to that. But for now, if there is anyone listening to this who would like to know more, who do you think would be the ideal audience for this to have, immediate takeaway actions to say like, okay, now I know I want to go and read this paper or get involved in this, healthcare policy movement?
I think some of the best work from a public health and a public awareness point of view that's been done in the UK is a charity called CMV Action. So CMV Action really are, I guess, a support and education group for mothers to be. So people who are having children may be worried about CMV, as well as parents who've had a child born with CMV. And so CMV Action is a really good starting point, and they do lots of great work in educating and informing people about CMV. I think if I had one lesson from all of this, I think it's the importance of asking the question of why something happens. And then the second question is, how does it happen? I think they're the two key questions that any scientist has to address. And it's as simple as that, actually. Obviously, you want to ask important questions, but never lose sight of that question of asking why something happens. So what we're interested in and what we've been doing is trying to understand how we can educate the immune response against a virus that infects us and can reinfect us despite the fact that we already have a really fantastic immune response against this virus. And by doing that, and by investigating that, we've identified that maybe what we have to do is educate the immune system differently. And by doing that, it may be better equipped to fight this virus. And we think this is really important because we think this could have impact on both our transplant patients and also to stop congenital infections.
Dr. Reeves, thank you so much for your time today.
Thank you.